Thank you, Robin, for the introduction. Hello, everyone. It's my pleasure to be able to speak to you about EIP Pharma over the next few minutes. EIP Pharma is a private clinical stage company based in Boston that is developing an oral drug that is the leading drug, disease-modifying drug for the treatment of dementia with Lewy bodies. In my presentation, I'll walk you through the science and the mechanism of action, which is on one level very specific to the pathology and targeted for the pathology in dementia with Lewy bodies, but actually has potential in a wide range of other indications as well. And then most of the presentation, I'll focus on the clinical data, particularly in DLB and our clinical program and plan going forward. A little bit more background on myself and the company and the management team. It's a highly experienced management team with C-suite experience in leading biopharmaceutical companies and a track record of developing and taking to the market transformative therapies in major disease indications. Also a board of directors that is primarily independent and is commensurate with that management team, and a world-leading top-tier scientific advisory board. So this is the pathology we're going after. Think of this as, instead of going after amyloid plaques, we're going after cholinergic degeneration in the part of the brain called the basal forebrain. This is a system that is involved in a wide range of regulating a wide range of cognitive and motor tasks. From a drug development standpoint, there are two reasons this is a particularly attractive pathologic target in neurodegenerative diseases to go after. One, it is very well correlated in a wide range of diseases to both clinical expression and disease progression, particularly in dementia with Lewy bodies, but again, in other indications as well. And more interestingly, in certain respects, from a clinical development standpoint, which is my background in particular, is that the neurodegenerative process in the basal forebrain, in the cholinergic system, is actually reversible. There's a wide range of preclinical data, animal data, as well as some human brain data that would argue why the cholinergic neurons actually don't die until very, very late in the neurodegenerative disease process. And because of that, you can actually reverse and improve function and reverse disease progression and not just rely on slowing disease progression overall. Now, one piece of data that we've presented very recently that actually goes along with this, which I think is actually quite interesting data, was presented by ourselves and our colleagues in the neuroradiology sector from the VU Medical Center at CTAD this past December. So this is looking at MRIs to assess volume of the major cholinergic neuron in the basal forebrain, called the nucleus basalis of minor, or NBM, and looking at the volume before and after 3 months of treatment with neflamapimod, our drug. The expected is that it's flat to a slight decrease, because in every longitudinal study, there is a decrease over time in this patient population, an early AD population. Instead, in the NBM, you see a median 4.5% increase, which is a significant increase, versus, again, the decline. And in the rest of the brain, in fact, you see exactly that decline of around a little bit under, around 0.5% over the 3 months of treatment. And this really goes along with the preclinical data and the mechanism of action I'll be showing you, that we can actually reverse the neurodegenerative process within the basal forebrain. So this is the pipeline. With neflamapimod, our lead asset, and dementia with Lewy bodies as our lead indication. So why DLB? So starting with a wide range of reasons, along with the clinical data, which I will come to, is that number one, there's less fixed deficit here. This is fundamentally a more treatable disease than Alzheimer's disease, particularly in the patients who have pure DLB. There is less frank neuronal loss in the cortex, and therefore the ability to reverse disease. Second, well-aligned mechanism of action in terms of the cholinergic degenerative process and the primary pathology in the disease, in DLB, which is in the basal forebrain. And third, high unmet medical need with no approved therapy, which along with reversibility means that you can go to the market with a 6-month phase 3 trial, rather than the 18 months and the cast of thousands that, particularly in the AD context, that you all know is required to be able to show effects on disease progression. And again, from a clinical development standpoint, what it means is much higher probability of translatability going from preclinical to phase 2, but particularly from phase 2 to phase 2b to phase 3, because you're only marginally extending the time or period, but the endpoint and the effect you're looking for is the same. You're not changing the goalpost in going from phase 2 to phase 3, which inherently, if you're chasing disease progression in phase 3, you are doing, as we all know, is the hard part of developing drugs in AD. So DLB, second most common neurodegenerative dementia after Alzheimer's, third most common neurodegenerative disease overall after AD and PD. High unmet medical need in every respect, stage for stage compared to AD, higher rate of cognitive decline, higher caretaker burden, more likelihood of institutionalization, et cetera. But there is therapy, there is a standard of care, which are cholinesterase inhibitors. Because it's a cholinergic deficit disease, to a certain extent, at least initially, they work better, but it is transient, and eventually there is a high unmet medical need for once a patient is stabilized on a cholinesterase inhibitor. And there is now a very specific diagnostic criteria, all of which, when you put it together, this is a very highly commercially attractive disease indication, the neurology call point, high unmet medical need, high pricing leverage. Put together, this is a great place for us and our drug to be in, particularly as we are positioned to be first in the market. One other comment to make here is that from a commercial standpoint, there's a nice development a couple months ago, a particular test called the DAT scan. It's a dopamine transporter scan, which had been approved to support Parkinson's disease diagnosis, was actually approved by the FDA to support a diagnosis of DLB as well. And within the data set, you can see that if you use a DAT scan along with that clinical criteria, your specificity against Alzheimer's disease is greater than 90%. So you can really now have a test that's approved, that's being used in the clinic to be able to specifically diagnose and manage patients with DLB. So I will not spend a lot of time on the science. I could spend a lot of time on the science. Our drug is a p38 alpha kinase inhibitor, as Robin mentioned. We licensed it from Vertex Pharmaceuticals, who were developing it in the classic fashion. P38 alpha inhibitors were being developed in the 90s and the 2000s as an anti-inflammatory for peripheral inflammatory disease. Vertex abandoned this drug because they recognized, and it was highly blood-brain barrier penetrant, which for rheumatoid arthritis, which was a disease indication, was not an optimal profile. It sat on the shelf. We licensed it out in 2014 when there was new science starting to come out with new biology that pointed towards the CNS. And obviously, that blood-brain barrier penetration ends up becoming a positive thing. In the literature, there are many different arguments for why you would want to develop a p38 alpha inhibitor in CNS disease, particularly for neurodegenerative diseases, focused around tau as a target and tau phosphorylation, as well as neuroinflammation. In the work we've done more recently and is published in this paper, working with someone named Ralph Nixon, who is one of the leading researchers in the neurodegenerative diseases field, we believe the convergence points for all of these, what's in the literature, is actually a protein called RAB5, which is involved in, and hyperactivation of which leads to disrupted proteostasis and endolysosomal dysfunction, all of which plays out in the cholinergic neuron. It's thought to be the primary basis for the cholinergic degenerative process. And we believe then that this is where and how our drug is primarily acting. Otherwise, we have a range of preclinical data, both mechanistic data with cellular potency consistently between 20 and 30 nanomolar, as well as in vivo animal data. And I'll just highlight again what is published in that paper, a physiologic neurodegenerative model of basal forebrain cholinergic degeneration, the TS2 mouse model, where we do see actual reversal with increases in the number of cholinergic neurons in the basal forebrain, as well as restoration of behavioral outcomes. So we are far advanced in development otherwise and ready for late stage development, because some of this is because Vertex had done the work in terms of toxicology, CMC, phase 1 clinical studies. Overall, now we have greater than 300 subjects of safety data, including at much higher doses where Vertex had gone to for rheumatoid arthritis, again, based on the different biology. And then we have data in prior studies in Alzheimer's disease demonstrating target engagement and blood-brain barrier penetration. And then in this study, and the results of this study, proof of principle data and clinical activity data in dementia with Lewy bodies. This was a phase 2A study, 91 patients, 16-week placebo-controlled study in a well-defined DLB population, including having had a DAT scan confirming the diagnosis. And the other patient population thing to keep in mind is that these were all patients who were on the current standard of care for their cognitive deficits, the acetylcholinesterase inhibitors. So the endpoints in the trial that we looked at are shown here. A cognitive test battery looking at the cognitive domains that are impacted in DLB. You won't see a memory test on there. That's Alzheimer's disease. These are the tests that you utilize to assess cognitive function in DLB. The CDR sum of boxes, I think many of you are familiar with that. And then the timed up and go test as a matter of motor function, which is very prominent, and deficits in which are very prominent in, particularly with respect to gait in DLB. So these are the overall results. In the ITT analysis, which included a low dose that was clearly not efficacious, the 40 milligram BID dose group. In that analysis, even with including that dose group, we see significant effects, improvement relative to placebo on the CDR sum of boxes and on the timed up and go test. Less effect on the cognitive endpoints, we believe, again, because they're on cholinesterase inhibitors already. But at the higher dose, the cognitive effect comes through as well, and particularly on the CDR sum of boxes, even a more robust effect at the 40 milligram dose group compared to placebo. Now, we did do some looking at trying to understand which population within the overall group are more responsive, and used a marker called of AD copathology called plasma p-tau 181, which measures is correlated to amyloid, both amyloid plaque and tau PET scan. But we believe what's most important with that marker is that it's a marker of the extent of neuronal loss and neurodegeneration, effectively fixed deficit that you can't reverse in a patient population. And if you stratify by a predefined cutoff of whether or not they have crossed that threshold for a tau pathology or amyloid pathology, we actually see very significant or substantial increase of effect on the major endpoints, effect sizes approaching large effect sizes. These are effect sizes that are rarely, if ever, seen in dementia clinical trials. Again, what it shows is that when you focus on the patient population that have a pure cholinergic deficit, you actually unmask and show what the full effect of the drug is, and not bound by the fixed neuronal loss in the cortex. Next step is to then do a phase 2B study. We could actually, with the data we have license from the FDA to go directly to phase 3, but we decided to do this in a staged way. we'll do a phase 2b study that takes the learnings from the phase 2a study, confirms that with larger patient numbers, and that will then allow us to go into phase 3, we believe, with largely removing any residual efficacy risk in going into phase 3. So the learnings that we're focused on are, again, one is finding the optimal dose, 40 milligram TID. Second, that the CDR sum of boxes, by being able to capture both cognitive and motor deficits, is actually a better endpoint than the cognitive endpoint that we had used as the primary outcome measure in phase 2A. And then third, again, focusing on the pure DLB patients to optimize outcome, but in the long run, delivering the most value on an individual patient basis, because these are the patients who have, again, the pure cholinergic deficit that our drug is best aligned with. So the phase 2b study that we are anticipating initiating within this quarter is a 16-week placebo-controlled study with 8-month extension. DLB in the early stages, CDR global score of 0.5 or 1, again, without elevated plasma p-tau181. So effectively a pure DLB patient population with a primary endpoint of the CDR sum of boxes, and then secondary endpoints that look at the other comprehensively the disease. We do also have a EEG marker that we had preliminary data in phase 2A where we actually did see a significant effect relative to placebo on beta functional connectivity. Because of COVID, we had the pandemic, we had reduced patient numbers. We haven't been touting this, but this is actually a biomarker we're going to try to confirm in the phase 2B study. And the study will be financed by a grant from the NIH that we are far into the process. And based on the scientific review and all the feedback, we will be receiving imminently. Pardon me? 2 minutes. Yep. Got it. So there are, again, as I mentioned, with the mechanism of action into the cholinergic system, there is other indications we could consider with appropriate financing. And we do have a co-crystal and patent around this with neflamapimod that gives us additional composition of matter, which may actually allow us to differentiate and go into some of these different indications. These are our upcoming milestones over the next 2 years now. Again, the next one being particularly around the phase 2B data, starting the phase 2B study. We do have a presentation upcoming at ADPD, which is more extensive presentation of the MRI data in terms of direct effects on the basal forebrain. But I think the most important here is that we're targeting third quarter of next year for the readout of the phase 2B study. Importantly, I think going on from here, it's not a huge phase 3 program, and it's actually very similar to the phase 2B study that we're looking at in phase 3. So that's part of why the phase 2B study result is significantly value creating, because you know that there's very little residual risk into going to approval into the market for for a major disease indication with significant commercial opportunity. And I'll just stop here. This is the long run. There's the opportunity to file an NDA with 2026 under this plan. Again, with financing that we can execute on this plan, and it's not the type of typical AD development program that one would be thinking about. Thank you very much.